Mattias?
Yes, I'm here.
I think you can start. Thank
Sorry?
you very much for being with us.
Okay. Thank you for the kind invitation, and thanks for hosting this event. I really like the set of people who are speaking, and I would have loved to be in Paris to share this day with you all. But this will have to do for the moment. So today, I want to speak to you about how we can get a better view of human history based on genomes, and in particular, benefiting from that we're able to sequence entire genomes, not only from people who live today, but also from people who lived in the past. So we are at a very exciting time in biology. We witnessed a remarkable development of being able to get DNA from or genetic information from multiple individuals, from maybe 20 years ago when the human genome was sequenced the first time, to having rather few bits and pieces of information from each individual, till today when we can sequence the entire genome of a few or many individuals in a relatively rapid and decently economical or affordable way. We're also fortunate in that a lot of mathematical people, population geneticists and statisticians were developing rich population genetic theory in the late 80s and early 90s. And this turned the perspective of population genetics on its heads, from having a forward perspective to actually having a backwards perspective, which fits very well with the type of data both from people who live today and people who lived in the past. When we have that type of data, these types of perspectives have been proven to be very, very powerful for making inferences about the past. As Ludwig was outlining earlier in his talk, we were also witnessing a very interesting and dramatic revolution in the ability to get DNA from ancient material, not only horse or human or mammals. This is going far and wide into sediments and into plant community, and I'm sure we have only seen the beginning of this trend. It's very exciting because we as evolutionary biologists, for the first time, we have the possibility of actually looking at the genetic material at various time points back in evolution, and not only be restricted to comparative type of analysis where you take a human and compare it to a chimp, for example. So when you are interested in human evolutionary history and the human past, if you start looking at what's been going on in the last 20 years, one thing become relatively apparent, that a lot of new and exciting results have come out in the last 20 years, many of them focusing on Europe or Eurasia or other parts of the world than Africa. Here's just an illustration from a review where you can see various types of results being presented on a map as arrows of migration, and the majority of them focus on the parts of the world outside of Africa. You can also see that many of these, or most of these events, are actually relatively recent. Yellow here means a recent event, and green means an older event. At the same time, we do know that humans, and in particular modern humans, evolved in Africa, in sub-Saharan Africa, some time ago. For instance, we can see that by comparing the chunks of inherited genetic material in human groups as a distance from Africa, for example. So on the x-axis of this little plot here, you have distance from East Africa, and then on the y-axis, you have the chunk, the size of these chunks of inherited material, and they grow larger and larger as you travel away from Africa. That's just one of the signals pointing to an sub-Saharan African origin of modern humans. We also know that if you go into the morphological record and you start looking into the human lineage or the modern human lineage, when you're hitting 150, 180, 200, 250, and 300,000 years into the past, the morphological diversity based on fossils is quite exciting and large in Africa. Obviously, there are other forms of closely related organisms like Neanderthals and Denisovans, also showing morphological diversity when you compare it to modern humans. But within sub-Saharan African the sub-Saharan African continent, there is an increasing level of diversity at this particular time point. Just to give us a bit of perspective, if we're going back in history in the sub-Saharan African continent, the people who were developing into modern humans were not alone. This remarkable result from the Homo naledi, for example, from the Rising Star Cave, pointed to this. So on the left-hand side here, you see a piece of fossil from Homo naledi. These were individuals and a species that lived in southern Africa. They lived just a few hundred kilometers away from another fossil find that resembles very much us modern humans from Florisbard, this particular one on the right-hand side. And the people on the Homo naledi, they're remarkable in the sense that they have feet like modern humans, but they have hands like apes, and they had a brain volume of about a fourth of modern humans. Here's just a reconstruction of these two different, based on these two different fossils, of what they might have looked like. And this points to that when we're going into deep time in Africa, there's going to be different types of Homo living at the same time. And there's no indication that these groups met and mixed in that sense and had fertile offspring. But there were probably many other forms of humans, archaic humans, if you call them other species, or this kind of transition landscape of different forms of Homo living side by side. The Homo naledi fossils have been dated to about 300,000 years old, and the Florisbard skull has been dated to 260,000 years old. So they were certainly meeting and on the landscape when the time was. So that puts us into a perspective of comparing to other parts of the world. So outside of Africa, we have fossil finds from Neanderthals and a little bit from Denisovans. And one thing that we're learning as we're going along with getting genetic material from Neanderthals and Denisovans is that the mixing with modern humans have been ongoing and quite a bit more complicated than we initially thought. But one very striking characteristic between Africa and outside of Africa is that we've had pulses of migrations of Homo out from Africa, and then probably long periods of no migration or no contact. So the ancestors of Neanderthals and Denisovans likely migrated out from sub-Saharan Africa perhaps 1 million years ago or on that time scale. And there were earlier forms of Homo migrating out at earlier times. But then for probably geographic and climatic reasons, there's been long periods of no contact between sub-Saharan Africa and the rest of the world. This makes finding and figuring out mixing between modern humans and archaic forms of humans like Neanderthals still complicated, but perhaps not as complicated as we can anticipate it will be when we dig deeper into the African record. So what do we know about human history in Africa from a genetics or genomics point of view? Well, if you look at the left-hand picture here, the map, there's a few distinct results that are emerging irrespective of what type of genetic data that you're investigating. It's quite clear that there's a relatively strong correlation between geography and genetics, especially if you look into indigenous groups of various parts of Africa. For example, if you compare the Khoi and San indigenous groups from Southern Africa to any other group, whether it's African or non-African, that's where you're finding the deepest divergences among all modern humans. In the central parts of Africa, we have forager groups, rainforest forager groups, also known as pygmy groups. There are not that many individuals remaining of these groups today, but they also show a relatively deep divergence among other groups on the sub-Saharan and the worldwide perspective. On the right-hand side, I show you some sort of reasonably well-known migration patterns, but they all date to perhaps the last 10, 15,000 years into the past. One event that will be important for what I'm talking about is a migration likely starting outside of Africa, getting into East Africa, picking up herding practices, and ending up in Southern Africa maybe 2,000 years ago. Now, another arrow here represents the Bantu expansion. This is probably the largest movement of people in the world, starting perhaps 4,000 or 5,000 years into the past, of people practicing first agriculture and iron working, spreading into East Africa and to South Africa, and in many places replacing or assimilating previous indigenous groups. So when we're interested about the deep human history, we may pose a number of questions, like when and how did early modern humans evolve? Is there a transition into modern? Is there a time point? Is there a genetic change that's different or important for this event to happen? Or is it more a gradual process? We can also pose the question, did modern humans emerge from one group, or did they emerge from multiple groups? And we can also ask the question of whether they mixed with archaic forms of humans. And we can think about these archaic forms of humans like Neanderthals, or at least distantly related to modern humans on the same level as Neanderthals. So now I'm going to talk a little bit about a project that started out on a very kind of non-deep divergence focused question. It started out from a relatively straightforward and simple type of task. So remember I showed you earlier that we have these distinct ancestries in different parts of Africa. And our purpose here in this first study was to try to figure out how long have the Khoisan, or the ancestors of the Khoisan, been living on the landscape in Southern Africa. How can you do that? Well, you can actually sample human remains that are a couple of 100 or a few thousand years into the past and figure out how similar are they to modern day Khoisan people. There are some people, human evolutionary biologists, who argue that our patterns of diversity we see today have very little resemblance with the events in the past, and that's been motivated by some of the events that we've been seeing in Europe during the Neolithic transition. So this project came about of looking into the genetics of a couple of individuals who were living on the southeast coast of Africa. And it turns out that we were able to get good DNA from a number of these individuals, despite that these individuals have been buried in very hot and humid areas on the south coast of South Africa. And for some of them, 3 of them are about, or died about 2,000 years old, 2,000 years ago. And for some of them, we could sequence the entire genome up to 13 times coverage of every base. Here's just an illustration of the different individuals. So the 3 top ones are died about 2,000 years ago. And then we have another comparative group, which are on average died about 500 years ago. And for some of these individuals here, we have good genome coverage. And for some other ones, we have poor coverage. Now I'm going to take somewhat of a technical detour. Or hang on. Let me first show you the results, or a very obvious analysis of these individuals, and then I'll take a technical detour. So one of the first things that you can do when you have this type of genetic data is to just figure out how genetically similar are these individuals who lived 500 years ago or 2,000 years ago. An obvious comparison is with other sub-Saharan African groups who live in sub-Saharan Africa today. So on the red here and the black dot here are the two of the 2,000-year-old individuals from the coast of South Africa. And our hypothesis was that these groups were genetically similar to modern-day San and Khoi groups. And in the first instance, when you compare it to the Yukoansi, for example, this is a San group, and another group in Southern Africa, the Kareki people, yes, that seems to be a straightforward answer, that the people or the remains from these people, they have a direct link to the modern-day Khoi and San groups. We compare them also to West African groups or to East African groups to get kind of a picture of the entire sub-Saharan African continent. But when you start staring at this figure a little bit more in detail, you realize that these ancient individuals, they are pushed further to the right on this first axis of genetic variation. And what we're viewing here is a representation of the genetic variation among the various individuals. And if you're pushed further to the right on this particular plot, that means that you're less genetically similar to the individuals on the left. And overall, if two dots that represent an individual are close to each other, that means that you're genetically very similar. So what can cause this pattern? Well, our first gut reaction was that this was an artifact, or this was something caused by the quality of the data from these ancient remains, but we could relatively quickly rule out that possibility. And there are a number of processes that can cause this pattern, but the very obvious one is actually admixture into the modern day groups. And if we start modeling this particular history, and if we're trying to model the Yukoansi, for example, a San group that live in Southern Africa today, the only way to model this modern day San group is to take, for instance, 88% of the ancient humans that lived there 2,000 years ago, and about 12% of the genetic material from an East African group. So this was an unknown result, and it fits well with what we know about the migrations from East Africa starting about 2,000 years ago. But we had no idea that this migration impacted, for instance, the San group. Now, this gave us a first kind of clear result. Yes, the ancestors of the Khoi and San, they were certainly living on the landscape in Southern Africa 2,000 years ago, but the people who live there today, they have been reasonably dramatically impacted by migrations in the last 1,000 to 2,000 years back in time. That also led us to pose another question. Previous studies have used the Yukoansi, for example, or San groups to date the deepest divergences among modern humans. And if you're doing that with a genome that's made up of 88% San in some sense, and then 12% from an East African group, which is then more similar to any other group that you're comparing to, you're going to get a biased result. So we decided to go after the question of what's the deepest divergence among modern humans, in particular because we probably have much better data with these ancient genomes of less impacted by admixture, but also since you can see the deepest divergence of modern humans as a lower bound of when modern humans must have been developed. Okay, so a quick technical detour. Since we're working with complete genomes these days, we can use very new and interesting tools of dating divergence times. So instead of what we've been doing in the past, when we've estimated some sort of population history of in a comparative set, we always had to factor in whatever potentially happened in the population sizes of the past of these two individuals. But with complete genomes, you can actually estimate directly the number of generations, irrespective of bottlenecks or any other type of events that happened in the past. And you can do that by just using the information in two completely sequenced individuals. And what you do is to extract out the sample configurations when one individual carry no derived, so no chimpanzee-like variants, and the other individual carry none of those derived, or only the ancestral variants, so only the chimpanzee-like. And then you have all these 9 different sample configurations, and that kind of really captures the essence of the model that I had on the previous picture. It also turns out that for these different sample configurations, all possible different events can then be derived so that you actually get a probability of observing a certain sample configuration. And the neat thing here in this equation system is that you have the parameter t, which is the number of generations that the two individuals in those two populations have been separated. So if you can estimate that, then you get to a divergence estimate that is not impacted by whatever happened in the history of those two individuals. So let's start with the sanity check. So if we use this method, and we can use another comparative method, the GFOX method, and we compare, for example, modern humans and Neanderthals and estimate the divergence times, then we end up with the now relatively well-known divergence time estimates on the order of 550 to 650,000 years into the past. If we take this ancient Southern African individual, the Balito Bay A boy, and compare it to any other modern human group, we end up in the time range of 260 to 350,000 years into the past. And that result seems to be very stable. We can put that onto a cartoon just to illustrate the divergence time in comparison to other human groups. So if we start outside of Africa, an estimate of the divergence to African groups is perhaps 80,000 years into the past. Then we have East African groups, we have West African groups, we have the Central African forager groups, and then the deepest divergence then goes back into 260 to 350,000 years into the past. And on the right-hand side, we have the San groups. This depth of time overlaps with, for example, the Flores bud skull that I already pointed out, that's coming or have been excavated in the very south of Southern Africa. It's also half the way or half the time back to the common ancestor of Neanderthals and Denisovans. So in some sense, it doubles the age of modern humans if you view this as a minimum bound on when modern humans must have been developed. I don't think it points to that this was the time of when every anatomical feature needs to have been fully developed, but I would say that it probably points to that the modern behavior or the things that we associate with modern human behavior was already developed at this time point. So this result also questions the idea of modern human behavior emerging quite late based on artifacts and artist work from, for example, the Blombos cave in Southern Africa. That's about 100 or maybe 80,000 years into the past. Okay, so let's just say one more word about the genetic changes that might be going on in this time period. So we might be interested in which genes that actually evolved rapidly in this time period, say around 300,000 years ago, or perhaps even earlier. You can do that by comparing modern humans to chimpanzee. You can do that by comparing modern humans to Neanderthals and Denisovans, but you can also do that by looking into any change that's common to everybody, all modern humans living today, because that's going to then predate that oldest event, that 300,000 event that I just talked about. So just to illustrate one exciting result from a couple of years ago, here is a type of analysis that tries to capture the signal of variants that are unique to the modern human lineage that all modern humans carry. And the stronger the signal, so the higher the value on the y-axis, the more distinct does it show selection prior to the diversification of modern humans, so prior to 300,000 years ago. And on the x-axis, you have the size of that region in the genome. And if I just highlight some outlier genes here that show up, I'll soon kind of summarize what I point to. But let's start with perhaps one of the most extreme gene regions, the RUNX2 gene region. It's almost like a full megabase of genetic material that's all modern humans almost completely carry derived variants. This gene has been found in comparisons with Neanderthals as standing out as being very different from modern humans and Neanderthals. And it's also a gene that's known that if you have mutations in this gene, you get a syndrome known as craniocaudal dysplasia. You get heavy eyebrow ridges, you get a heavier skeleton, you get a rib cage which is flared, more similar to a Neanderthal-like rib cage. So basically, this gene has to do with morphology. A number of these other genes also has to do with morphology. There's one gene that has to do with the closing of the fontanelle that shows also heavy evolution in the early modern human lineage, and other genes also involved with cartilage. So kind of a rhetorical question would be, why would morphology be important for the development of modern humans? And I don't have a very good answer to that. All I know is that many of these genes are also deeply involved in cognitive functions. So 3 out of the top 5 genes show signals for morphological change, but also for cognitive change. Okay, so I'm going to try to wrap up here. I think what we're witnessing is a very exciting time when there's a lot of new results coming out, but there's also a lot of new questions coming out. So for instance, I've talked about this fluorspot skull, and I've talked about the deep divergences between the Khoisan San and the ancestors of the Khoisan San to everybody else, pointing to Southern Africa being an important part for human evolution. We know from the fossil record that East Africa was also likely a very important part for human development. And we also relatively recently learned from Jean-Jacques Heublin's work of dating the Jebel-Irhoud skull. This is an exciting skull that has many modern human features, but also some archaic features. And that's dating into that 300,000 horizon. So to summarize that, it points to that there are many places in Africa that might have been important for the development of humans, and that we actually know quite little about where, when, and how modern humans arose. And just to connect us back to where I started, even though modern humans developed from perhaps a diverse gene pool across the African continent, there were certainly other Homo lineages on the landscape at the time, possibly interacting with the people who then emerged into modern humans. So with that, I'll end. I'll thank Carina Schlebusch, who's a group leader here at Uppsala University now. And I'll also thank Marlies Lombard, who's a long-term collaborator on working with South African human remains. And then thanks for funding, and thanks for your attention.